Good evening, everyone. So today I'll be talking about LCMS-based lipidomics to understand cholesterol metabolism in mammals. So to begin with, I'll just give a brief intro of what lipids are. So lipids in general do not have any standard definition as such, although some people do characterize lipids as those molecules which are soluble in organic solvents and are insoluble in water. However, this definition is not very normally used, and the more acceptable form of definition is that lipids are fatty acids and their derivatives are substances that are related to these forms. So when we say lipids, for most people, the most immediate thing that comes to their mind is the cellular membranes, which are composed of phospholipids. However, lipids have a lot of different functions. So the first function of lipids is to maintain structure in the form of glycerophospholipids or cholesterol. Lipids are also used in terms of maintaining storage, where they are stored in the form of fat droplets or lipid droplets inside cells. And some of the lipids which are involved in this process include triacylglycerols as well as cholesterol esters. And the third function of lipids includes signaling, where lipids bind to different protein partners and can regulate different downstream signaling events. And lipids like sphingolipids or endocannabinoids do a lot of these processes. So for my talk, I'll be focusing on one particular class of lipids known as cholesterol and cholesterol ester. So cholesterol is a ubiquitous lipid that is present in all cells in mammals, and generally there is a tight regulation of cholesterol in general exists in two major forms. One is the free form, which is used for maintaining the membrane fluidity in cells, as well as it is used for formation of lipid rafts to transport different cargo within cellular or within cells. And it is also the precursors for different hormones as well as bile salts. And this cholesterol is generally converted to fatty acid esters known as cholesterol esters, which is shown here, by two major enzymes, which are ACAT and LGAT. So the substrates for these enzymes are long-chain acyl-CoAs or phosphatidylcholine. And these cholesterol esters are majorly used lipid droplets, or they are used for transport of cholesterol within the body in the form of lipoprotein. And these cholesterol esters can be converted back to free cholesterol through the action of enzymes known as cholesterol esterases. So, physiologically, the concentration of these molecules is very important since they are associated with various different disease pathologies. And over the years, there have been several different techniques to measure cholesterol. And one of the oldest techniques is known as the Abel-Kendall method, which is a colorimetric method where the total cholesterol is measured. Here, cholesterol esters are first converted to cholesterol through saponification, and this cholesterol is converted to cholesterol-hexane sulfonic acid through the action of acetic anhydride as well as sulfuric acid. And this generally has an absorption maxima at 410 nanometers. However, this method, although rapid, it's not very qualitative or quantitative in nature for the cholesterol ester species, and you don't get to know any sort of diversity for these molecules. The advancement of mass spectrometry has yielded to the development of GCMS and MALDI-based MS techniques to analyze cholesterol and cholesterol esters, and these techniques generally involve the derivatization of both these molecules to separate and measure them. Due to the derivatization process, it also involves a lot of sample prep, and it may lose on the original structure of the molecules. Lately, lipidomics have been emerged as an important tool where LCMS-based approaches have been applied, and cholesterol measurement also is one of the most important approach which people are trying to do. So the challenges in cholesterol and cholesterol ester measurement using LCMS-based lipidomics is that these molecules are generally poorly ionizable, and to circumvent this, people have tried to use direct infusion techniques or try to derivatize these molecules in order to ionize these in a better manner. However, since these molecules are also hydrophobic, they are very difficult to separate in using normal reverse phase chromatography, which is usually used for general lipidomic protocols. And due to this, people have adapted normal phase chromatography with hexane or chloroform, and this generally yields a good separation, but there are certain disadvantages with the following approaches which are mentioned here. Both these approaches include a lot of cumbersome sample preparation techniques, And these involve involve standalone LCMS instruments because these molecules during direct infusion or use of corrosive solvents like hexane generally requires a lot of support, and they include a lot of system breakdowns to bridge the gap of to develop a simple LCMS technique. We wanted to measure cholesterol and cholesterol esters. So for this, we use two different standards which are commercially available. The first one is a deuterated form of cholesterol known as cholesterol D7, and the second one is cholesterol heptadecanoate, which is a 17-chain fatty acid containing cholesterol ester. So we initially injected 1 nanomole of both these compounds in a C18 column, and we optimized the solvent system to separate these molecules in a good manner. So using the solvent system as shown in the slide, we could separate both these standards to a very good extent, and both these molecules were observed in a good intensity. So once we separate these molecules, we wanted to confirm these molecules' fragmentation pattern as well. So with the current solvent system, cholesterol generally forms an ammonium adduct, which is quite unstable, and even without the application of any collision energy, it dissociates to form a dehydrated cholesterol variant. So this dehydrated cholesterol generally forms the precursor ion, and this cholesterol molecule can be further fragmented on the application of collision energy. When we applied collision energy of 5 volts, we could see that we could get a good pattern of fragmentation where known reported fragments of 256 and 283 of cholesterol was observed using the LCMS method of ours. Similarly, for cholesterol esters, without application of collision energy, the ammonium adducts that are formed are pretty stable and they are detected as precursor ions. And with the application of collision energy, the fatty acid chain breaks off to form a dehydrated cholesterol, which yields a fragment at 369, which forms a signature fragment of all the different cholesterol ester species. So once we have established that our method can be used to separate these molecules as well as fragment them in a good manner, we wanted to identify our sensitivity of our method. So for this, we basically injected a mixture of both our standard molecules with different concentrations, and we could see that we can have a dynamic range of over 3 orders of magnitude, and the limit of detection for cholesterol was around 10 picomoles, and the limit of detection for 17,0 cholesterol ester was around 1 picomole. And these amounts are much, much lower than what was already reported for these molecules using existing techniques. So after this, since we have a established LCMS method for these molecules, we wanted to profile these molecules in cell lines as well as mouse tissues. So for this, we utilized the following workflow where we took cells or mouse tissues and we homogenized them and extracted the lipid content from these cell lines or mouse tissues using chloroform methanol. So once we extracted the lipids were subjected to mass spectrometry using an untargeted approach in a Q-TOF mass spectrometer. And after this, we analyzed the data where the area under the curve for each of the different species was normalized to the internal standards as well as the protein content or the tissue weights of the respective sample. So we chose 3 cell lines for this study because since cholesterol is important for immune and nervous system, we chose these 3 cell lines. HEK serves as a general mammalian cell line, and raw cells serve as an immune cell line or a macrophage cell line, and N2A cells serves as a neuronal cell line. So we measured the cholesterol content in these cells using our method, and we could see that raw cells possess the highest amount of cholesterol compared to the other 2 cell lines. And this was not surprising to us since generally immune cells require a lot of cholesterol to orchestrate different immune processes. And due to this, the amount of cholesterol is typically in a high amount for these cells. After this, we profiled the different cholesterol ester species, and the graph here basically shows a heat map of different species of cholesterol ester of varying chain lengths with different unsaturations, which are plotted to the mean concentration that are normalized to the protein content of the cells. So from this heat map, we can clearly see that all the 3 cell lines And this are the species of cholesterol, say 18.1 and 18.2. And even among these species, we can see that there is a preferential distribution for 18.1 and 18.2 between raw and N2A, where raw cells have 18.1 as the highest abundant species, whereas N2A have 18.2. However, the preference for these species is not known to our end. So, after profiling the cell lines, we wanted to profile the different tissues and mouse blood for cholesterol and cholesterol ester. And for this heat map basically shows the different cholesterol ester species and cholesterol that are profiled for different mouse tissues that are shown here. And this heat map is basically plotted in a logarithmic scale to the concentration of the cholesterol or cholesterol ester species. And from this heat map, amount, and these three species are 18.1, 18.2, and 24. So from here, we can see that the unsaturated species or the polyunsaturated forms of cholesterol ester species of 18.2 and 24 were the most abundant in all the tissues, and 18.1 and 18.2 were also abundant in cell lines. So among cholesterol, we could see that brain had the highest amount of cholesterol, and this was falling into place with the already known literature that de novo cholesterol synthesis in brain was one of the highest among all the tissues in mouse. Further, we could also see that blood had a great diversity of cholesterol esters due to the heightened activity of LCAT in blood, and cholesterol was lower in blood in general because cholesterol is actively converted to cholesterol esters through the activation of LCAT in blood. So right now, we know that we have profiled the different species. We also wanted to confirm that our method is capable of identifying changes in cholesterol and cholesterol esters when metabolic changes occur. So for this, we basically used orlistat, which is a general lipase inhibitor, and this also is used as a commercial drug for treating obesity. So we used this drug and treated it with raw cells to inhibit any active cholesterol esterases if present in these cells, and if this is done, we could see a clear increase in cholesterol ester levels. And here I am showing a representative image of C18.1 cholesterol ester, which is one of the most abundant cholesterol ester in raw cells. And here we could clearly see that after treatment with orlistat, the 18.1 species of cholesterol ester increases, whereas cholesterol decreases, showing that our method is capable of identifying changes in cholesterol and cholesterol ester levels with perturbations in the metabolic level. So with this, what we try to do with our study is that we have established a LCMS method which is simple as well as robust to tandemly measure both cholesterol and cholesterol ester species, and using this method, we can couple with different pharmacological screens or genetic screens to understand cholesterol metabolism in a much deeper extent, since the cholesterol esterases, which are part of the cholesterol ester metabolism, are not well known and they remain cryptic in nature. And this method can be extended to understand those roles in a much deeper manner. Further, this LCMS method can also be extended to clinics or pathology labs to measure cholesterol and cholesterol ester, since overall in the world, people generally measure cholesterol, however, cholesterol ester levels still remain unknown. And it is highly important to measure these molecules since both these molecules play a huge role in different disease pathology. Further, we can also modify this current method to identify different immunologic lipid mediators like oxysterols or oxidized cholesterol ester, which are highly potent molecules that occur during oxidative stress. So with this, I would like to thank my supervisor, Dr. Siddhesh Kamath, for the opportunity and all the lab members who are present for all the support and the IISER mass spectrometry facility. And I would also thank biocrates for the opportunity to present my work here.